Hello everyone, let's talk about PCR, which stands for Polymerase Chain Reaction. It's a laboratory technique that's used to amplify and detect a specific segment of DNA or RNA. This technique is highly sensitive and specific, which means it can identify even small amounts of genetic material, like viral or bacteria DNA in clinical samples. Now, let's break down the steps involved in PCR. There are three key steps: denaturation, annealing, and extension. These steps work together to create millions of copies of the target DNA or RNA sequence. Once this amplified material has been created, it can be detected using various methods, such as gel electrophoresis, fluorescent dyes, or probe-based assays. So, what are some of the clinical applications of PCR? Well, it's commonly used in clinical diagnosis to identify pathogens responsible for various diseases, such as infectious diseases caused by viruses, bacteria, or fungi. It's also used in genetic testing for inherited diseases, as well as in cancer diagnosis and monitoring. Let's take a closer look at some specific examples of clinical applications of PCR. First, it can be used to detect viral infections, such as HIV, hepatitis B and C, and COVID-19. Second, it can help identify bacterial infections like tuberculosis, meningitis, and sepsis. Third, it can diagnose genetic disorders such as cystic fibrosis and sickle cell anemia. Fourth, it can detect cancer-associated mutations in tumor DNA. And finally, it can be used in forensic analyses like in DNA profiling and paternity testing. The PCR test works on the principle of amplification of genetic material, which means that even tiny amounts of DNA or RNA can be detected. To carry out the PCR test, several steps are involved. First, the genetic material is extracted from the sample and purified to remove any contaminants. Next, short segments of DNA or RNA called primers are designed to match specific regions of the genetic material of interest. These primers are added to the sample along with other components such as DNA polymerase enzyme and nucleotides, which are the building blocks of DNA or RNA. The PCR machine then heats and cools the sample repeatedly in a process called thermal cycling. During each cycle, the DNA polymerase enzymes copy the genetic material between the primers, doubling the amount of DNA or RNA with each cycle. This leads to a rapid and exponential increase in the amount of genetic material, which makes it easier to detect and analyze. Once the genetic material has been amplified, it is detected using various methods, such as fluorescent dyes or probes, which bind to the specific genetic material being tested. By analyzing the presence or absence of the genetic material, it is possible to determine whether the sample is positive or negative for the target organism or condition. Let's take a closer look at how PCR is used to diagnose human papillomavirus, or HPV infection, which is a common sexually transmitted infection that can lead to cervical cancer in women, as well as other types of cancer in both men and women. To diagnose HPV infection, a sample of cervical cells is collected from a woman during a routine Pap test. The sample is then sent to a laboratory where PCR is used to detect the presence of HPV DNA. The PCR process involves amplifying small fragments of DNA in the sample using specific primers that target the HPV DNA sequence. This allows for the detection of even very small amounts of HPV DNA in the sample. To detect the amplified DNA, fluorescent dye that bind to the DNA are used. If the PCR test is positive for HPV DNA, the woman may require further testing, such as colposcopy or biopsy, to determine if the infection has progressed to a precancerous or cancerous stage. It's important to note that PCR is a highly sensitive and specific method for detecting HPV infection, making it an effective tool for cervical cancer screening and diagnosis in clinical settings. The PCR reaction is run in a thermal cellar machine, which provides a series of temperature cycles that are optimized for the PCR reaction. The thermocycler heats and cools the reaction mix to specific temperatures, providing the necessary conditions for DNA denaturation, primer annealing, and extension of the PCR product. The PCR reaction typically consists of the following steps. 1 is denaturation. The thermal cycler heats the reaction mix to a high temperature, usually around 95 degrees Celsius, which causes the double-stranded DNA template to separate into 2 single strands. Second is annealing. The thermocycler cools the reaction mix to a lower temperature, usually around 55 to 60 degrees Celsius, which allows the primers to bind to the single-stranded DNA template at specific locations. Third is extension. The thermocycler heats the reaction mix to a moderate temperature, usually around 72 degrees Celsius, which activates the polymerase enzyme. The polymerase enzyme synthesizes new strands of DNA using the DNA template and the primers as starting points. This process results in the amplification of the target HPV DNA sequence. 4 is repeat. The thermocycler repeats the denaturation, annealing, and extension steps multiple times, usually around 30 to 40 cycles, to amplify the target HPV DNA sequence. Gel electrophoresis. One way to analyze the PCR products by running them on an agarose gel using gel electrophoresis. The agarose gel is stained with a DNA intercalating dye, such as ethidium bromide or cyber green. These dyes bind to the DNA and fluoresce under UV light, allowing the DNA fragments to be visualized. The presence of a specific DNA band of the expected size on the gel indicates the presence of HPV DNA in the sample. The size of the band can be estimated by comparing it to a DNA ladder, which is a mixture of DNA fragments of known sizes that is loaded onto the gel alongside the PCR products. Gel electrophoresis involves the use of a gel matrix that contains small pores or holes through which DNA fragments can pass. Agarose, a polysaccharide derived from seaweed, is the most commonly used gel matrix for DNA separation. The agarose gel is placed in a horizontal or vertical electrophoresis chamber, which contains electrodes that generate an electric field across the gel. The PCR products are loaded into the wells or pockets that are created in the agarose gel. The wells are usually made with a comb that is inserted into the gel before it solidifies. Once the PCR products are loaded into the wells, an electrical signal is applied to the gel. The DNA molecules in the PCR products have a negative charge due to the phosphate groups in the DNA backbone. As a result, the electrical field causes the DNA molecules to migrate towards the positive electrode. The DNA fragments are separated based on their size, with smaller fragments migrating more quickly through the gel than larger fragments. The separation is due to the resistance of the gel matrix, which slows down the movement of larger DNA fragments. The time required for the DNA fragment to move through the gel depends on the size of the fragment and the concentration of the agarose gel. The interpretation of results. Finally, the results are interpreted. The presence of HPV DNA in a sample indicates a positive test result, and the absence of HPV DNA indicates a negative test result. The HPV genotyping can also be performed by sequencing the PCR products using Sanger sequencing or next-generation sequencing technology. Taking together, PCR is a technology that has a wide range of applications in vitro diagnostics, or IVD. The primary purpose of PCR is to detect and quantify nucleic acids in various samples. This technology has several characteristics that make it a popular choice in IVD. Firstly, PCR is highly sensitive and can detect even the lowest levels of nucleic acids in a sample. This sensitivity makes it an effective tool for identifying infectious diseases or genetic disorders. Secondly, PCR is highly specific, meaning it can distinguish between closely related nucleic acid sequences. This specificity is important for accurate diagnosis and identification of pathogens. Thirdly, PCR is highly reproducible, meaning it can produce consistent results from one experiment to another. This reproducibility is crucial for clinical use, as it ensures that results are reliable and consistent. Fourthly, PCR is speedy, with typical experiments only taking a few hours. This speed allows for rapid diagnosis and treatment which can be life-saving in some cases. Fifthly, PCR can be automated, which minimizes the risk of human error and allows for high-throughput testing. Finally, PCR is highly flexible and can be adapted for a variety of different applications, including genotyping, pathogen detection, and quantification. In summary, PCR is a highly sensitive, specific, reproducible, speedy, automated, and flexible technology that is widely used in IVD for the detection and quantification of nucleic acids. Then, we will be exploring the different products available on the market for PCR-based diagnostic systems. The first example we will be introducing is the COBAS PCR systems. These are automated platforms that utilize PCR technology for the detection of various infectious diseases, such as HIV, hepatitis B and C, and HPV. These systems are highly effective and have been widely used in clinical settings. The second example is the Gene Expert systems. These are also automated PCR-based diagnostic systems that can detect a range of infectious diseases, including tuberculosis and HIV, in just a few hours. This rapid turnaround time is particularly useful in emergency situations. The third example is the Anyplex 2 multiplex PCR assays. These assays are used for the detection and identification of multiple pathogens in a single reaction. This makes them particularly useful for epidemiological surveillance and outbreak investigations. Finally, we have the AccuPower PCR premixes. These are ready-to-use PCR reagents that simplify the PCR process and reduce the risk of contamination. These premixes are particularly useful for those who are new to PCR or those who need a quick and simple solution. It is important to remember that the choice of product will depend on the specific diagnostic needs and requirements of the user. Therefore, it is always important to consult with a qualified medical professional before using any diagnostic product. Thank you for joining us today, and we hope you have gained a better understanding of PCR technique for IVD.